For those of you who don't know me,
It's just rough. The whole car is just rubber.
so for those of you who don't know me, my name's Jonathan Mitchell. I'm doing a postdoc at the University of Alaska Fairbanks, but due to COVID, I'm back living in Hobart. So yeah, so this session is the last one of Phylomania 2020. It's on stochastic models of evolution, and we've got 5 live talks, all from University of Tasmania. And the first one is going to be a joint talk by Malgorzata O'Reilly, who's on Zoom, and Barbara Holland, who's here live. So take it away, Barbara and Malgorzata.
Thanks, Jonathan.
Thank you.
Yeah, so maybe just do a voice check, make sure we can both be heard. I think we can. Yeah. So, yeah, so I'm Barbara. Margajata's the one on Zoom. So the last, I guess, few years now, we've been enjoying working together, sort of trying to basically uncover some of the ways, some of the tools that Margajata has from this world of matrix-analytic methods can be applied to various problems in evolution. And so our talk has promised sort of species tree, gene tree reconciliation. In fact, most of it will have a section where we talk about some of the models we've been thinking about for species trees, and then a section on gene trees, and we'll just briefly mention the reconciliation. Over to you, Margajata.
Okay, so matrix-analytic methods is this wonderful, rich area of tools of applied probability, which goes well beyond 4-state Markov chains, and it allows for modeling a wide range of various real-life processes. And as Barbara said, we have been working together, talking about how to bring those modern tools of applied probability to the area of phylogenetics, and conversely, how to enrich the area of applied probability with the ideas from phylogenetics and construct new tools and new models and new ideas.
Yes.
Over to
you. So I guess, yeah, we put ourselves first because we've got a couple of our students will be coming up talking. So the piece on species trees, you'll hear a lot more from Albert, and then the piece on gene trees, you'll hear more from Jahal, and you won't hear from Tristan on reconciliation, but he is working on it, but he's going to talk about something different. Right. So you wanted to say just a little bit of background here, Margajata?
Yes. So matrix-analytic methods is an area that was pioneered by Professor Marcel Newt, who was keenly interested in creating probabilistic methods that will be applicable. So rather than having some wonderful integration or some other pure maths type of expressions in the theory of probability, he was very keen to develop methods that use matrices, for example, so we can compute them using MATLAB as an example, and algorithms, and basically the idea was to be able to compute things using computer. I had an honor of meeting Professor Marcel Newt at the time, and he's always been interested in technology. I had an honor to become his Facebook friend even, and so this area is really rich and interesting. And through this talk today, I'll provide some references to various literature that is interested and relevant to the topic today.
Cool. So just sort of brief overview of who we are. So we've already introduced the two of us and mentioned Jahal and Albert, who are both doing PhDs, and Tristan, who's doing a postdoc. So 2 other people, unfortunately, I think can't be here today, but have been very important in the group are David Livelez and Amanda Wilson. So they're both based in America and have decided to enjoy Thanksgiving and it being the middle of the night as well, I guess. But yeah, so they're on the biology side of things and have sort of kind of tried to keep us a little bit grounded in biological reality, which has been very useful. And so one of the things David Limmel was always sort of banging on about is that he would like the models that get used to be sort of mechanistic to the greatest sort of possible extent that they can, because that will provide, I guess, better testable hypotheses and predictions. And so that's sort of one of the philosophies that we have been, I guess, adhering to. And so what we've been basically, I suppose, trying to do is inject a bit more, I guess get closer to mechanistic models, both in terms of thinking about modelling speciation and extinction, but also for gene trees, sort of trying to sort of use some of this work of the sort of sub-functionalization model to think about sort of duplication and loss events and how and yeah, so rather than just having like your good old exponential distribution sort of survive, describing every sort of waiting time to a speciation or extinction or a duplication or a loss, sort of think, well, what are some of the actual constraints that are acting on these processes, and what sort of new distributions does that lead to? So beginning with the species tree side of things, we've been basically exploring these Markovian binary tree models, and these are sort of pretty flexible. So essentially you have this idea of an underlying phase or kind of a state that a branch might be in. The phase might depend on the phase of your parent branch, but rather than I guess your sort of typical phylogenetic assumption where you're always in exactly the same. If you have a speciation, your children are always in exactly the same state. It sort of can sort of generalize that. So we still have for our species trees the assumption that speciation implies independence. So once you've speciated, you're kind of doing your own thing, or conditional independence, perhaps is a better way of saying it. And so two kind of models of speciation that have been talked about quite a bit in the literature in phylogenetics are the sort of a symmetric mode of speciation, which is basically, so you have a speciation event and then the two children of that are essentially identical to each other. And that's sort of depicted on the, what is it, the left for everyone, yes. And your other model is this sort of asymmetric model of speciation, where you kind of think of a parent just carrying on after a speciation event and a child budding off. And yeah, so those are the sort of two main models, but these MBT sort of framework, I guess, allows you to do richer things as well. So I've alluded to the fact that usually in phylogenetics, if there's sort of some underlying state at a speciation node, that just gets copied to both the children. But here you could have speciation potentially being associated with a shift in phase. And so I thought about this with Arndt's talk, obviously, yesterday, sort of thinking about these cells sort of diverging. So like there's sort of like potentially kind of broader applications there that fit within this framework. So a Markovian binary tree, yeah, so you've got essentially a Markov chain describing a birth process, but you've also got this underlying phase variable that's sort of recording some extra information. And so the way these things have been sort of set up is you basically kind of define matrices that sort of tell you what are your birth rates, what are your extinction rates, but also some sort of extra structure that says, okay, when I speciate, what happens to the phase of my children? And so so you don't have to use that. So you can so for example, actually Margajada, this slide doesn't have a green dot. I think you were going to talk about it. Do you want to jump in?
Yes, yeah. I wanted to mention some things also. So the sort of ways that the Markovian binary trees, but also various other models from the theory of matrix-analytic methods, where they allow you to model a lot more than just assuming Poisson rates. So a lot more than just assuming that time spent in some state is exponential. And so Markovian binary trees is such an example. Now, a simple example of a Markovian binary tree is a BC model, where essentially you have two underlying phases, 0 and 1, where mu0 and mu1 are phase-dependent speciation rates, lambda 0, or is it the other way around? Lambda 0, sorry, lambda 0, lambda 1 are the speciation rates, and mu0, mu1 are the extinction rates that depend on the underlying phase being 0 and 1. And also you have this Q0, Q01 and Q10 rates of transitions between the phases 0 and 1. So this is what you will read when you read the paper on the BC model. And then what we notice is that you can actually frame this BC model as a Markovian binary tree with the following parameters. And so in that sense, BC model is some special example of the MBT.
Yeah.
And similarly with the MUSI model, so that's another example of a Markovian binary tree. And essentially you will notice that in this matrix P, we have ones and zeros. So some entries are set to 1 because at the moment of giving birth, a parent essentially does not change the phase, and the phase of the child is the same. And so this is again some sort of simpler form of Markovian binary tree, which can of course model a wide range of other behaviors. And a note here that I'd like to add is that in fact Markovian binary trees who were I guess introduced first in 2006 by Nectarius Contoleon in his PhD thesis, in fact are a special example of some other type of models. So for example, quasi-birth and death processes were introduced by Marcel Newts in 1981. So there's a vast literature on this type of models, and in fact Markovian binary trees and so BC model and MUSI model can be analyzed efficiently using quasi-birth and death processes. And also it's a special class of continuous time multi-type branching processes which were introduced in 1972. So there is a vast literature of various results and algorithms, very, very interesting and potentially applicable to the phylogenetics analysis. And so this is what we're doing in this work. I'd like to make an important distinction between a true tree, which is the tree that if you could imagine that you have all data available, you know precisely everything what happened, all the times and speciation events, extinction events. So the true tree records all information. And reconstructed species tree is what we have based on the actual available data. So reconstructed tree is missing some information. So here what I'd like to point out that in the calculations for the likelihood of a reconstructed tree, we really need to pay attention to the fact that there could have been some speciation events that ended up with extinction events that we do not know about. So in this right, the reconstructed tree on the right, so T star is the reconstructed tree, W is W, Z, and T is the times that we know of. However, you will notice that there were some speciation events that ended up being with extinction events, and we do not know about that. And so we introduced this notation G, W, Z, T, I, J to correspond to a likelihood of observing this internal branch that started at time W, ended at time Z with some speciation events, and T is current time, and I and J is initial and final phase. And also we introduce new notation D1, T minus W for the likelihood of observing this external branch that started at time W and is now observed at time T. Some measures that we have derived using matrix-analytic methods and some expressions and algorithms include the likelihood of observing a reconstructed T. We also have expressions for the likelihood of observing a true tree if you were to know all the information. We also are interested in some balance metrics, and so we have expressions for what we call DI given NT, which is probability time dependent probability that if at time T you observe a tree with N tips, I tips will be on the left subtree. And also we're interested in the limiting limits of that, so limiting probability. We have derived various measures. So, for example, for the likelihood, which we derived under general Markovian binary tree model, we derived various differential equations, various algorithmic sort of iterative expressions. But perhaps as a simpler example, let me present this formula for likelihood under the variant number 1, which is essentially each child is a similar copy and starts according to some initial distribution. And so you will see that in the expression for the likelihood, you see those earlier introduced notations because we have those internal branches whose likelihood we'd like to compute, and also external branches. And an example of variant 1 model is in an upcoming paper by Albert. How we derive the various results? Well, we use differential equations in various iterations. To solve differential equations, you can just use MATLAB solver. There's inbuilt MATLAB solver for that. However, differential equations are notorious for having various issues with instability. So we also explore various iterations. In particular, this measure of interest, the matrix of interest, is this DI given NT. So that that is something that we have derived expressions for. And here is an example of some differential equations that we use. Equation number 3 is well known. And equation number 4 is something that we have derived amongst some other expressions of sort of similar forms. And for equation number 3, there exist some methods that you can use. With the reference is provided below. Okay, and so what we are interested in is the balance matrix of a tree. Traditionally, when given some set of data, you might use this formula, which is expressed in terms of gamma function and beta parameter, which is some sort of way that you can approximate this probability of observing I tips on the left subtree in a tree with N tips using statistical methods. And so what we did is once we calculated our DI given N values, we then try to fit parameter beta to see, how close we can get the shapes of the two functions. And here are some examples of application of these methods. Now, this is work in progress. So to produce this graph, I just applied inbuilt MATLAB solver, and we're going to also explore some other perhaps more stable ways of doing it. But anyway, you can see that the output that we get here is something that we expect, because we have this sort of symmetry. So we expect that beta will be, the parameter beta will be approximately 0. Well, actually it should be 0, but because it's a limiting probability, we in fact calculate this DIN given DI given NT values for some large T. So there's still some convergence going on.
Yeah, but should point out, so the scale on the y-axis there, like, so for a sanity check that this should be essentially flat. It's not too bad, like we're kind of...
Yes. Yeah, that's right. So we do...
So this is a busy model where the speciation and extinction actually don't differ by space, so state. So you're expecting a uniform ranked tree topology, sort of basically the Yule style, constant birth death style distribution of tree shape. So we are sort of getting there. Very pretty. And this one here is just my attempt to try to evaluate things recursively, so there's some convergence of the values, time-dependent values, so extinction happening before time t, and so that's the graph of that, and you can see that E0, which is probability of extinction, given that you start in phase 0, is equal to 0.3, and so is E1. So we record these values also. Maybe Barbara, if you can take over the rest of the examples yeah, well, I think we're running a little low on time, so we'll just sort of say, so some of the things that we are sort of interested in is if we have a sort of an asymmetry in the Bissey model, do we get tree shapes that don't have this sort of standard balance you get out of a constant birth death model? And yeah, so we do see some examples where we can get shapes that are different, and we also see some examples where we get shapes of this sort of distribution of number of things on the left that actually aren't captured well by that beta splitting distribution introduced by Alder. So you're getting kind of a broader class of sort of tree shape distributions. I'm going to zoom right through all of these, given we've been told we have 5 minutes to go. You can just believe me that we've got lots of interesting examples. And just quickly say a little bit about the gene tree side of things as well. So one of the interesting distinctions in the models of gene trees we're looking at is we're thinking about genes have functions, and that those functions are protected by selection. And so what this means is it actually introduces some interesting non-independence. So if you lose, one gene, that might mean that you can't lose another gene. And so unlike the situation with the species tree, you don't have this independence built in. And so Jahal's been looking at QBDs to model gene tree evolution. And so in a QBD, you have a level and a phase. So for us, the level is just the number of genes in the family, and the phase can be sort of some potentially complicated variable. So in our case, it's recording some information about what genes can do what functions, which then also helps you know what genes can potentially be lost. And so the QBD gives you some nice structure in that the level variable can only change by one thing at a time. You either lose a gene or you gain a gene. You can't go instantly from 2 genes to 10 genes. And so that gives you a rate matrix, this sort of block diagonal structure where the blocks are moving you between levels, and then the matrices within a particular block are sort of changes of phase. So maybe the gene loses the ability to do some particular function. That would be a change of phase. And so Jahal has been, again, we've still been interested in this idea of, what can we learn about tree shapes of gene trees? And so he's sort of set up a model that records number of genes, number of redundant genes, and how many of each of those are on the left. And then we have some sort of underlying parameters of our model. So these mu's down the bottom that are essentially what's the rate you lose a coding region, what's the rate a gene duplicates, what's the rate you lose a regulatory region, what's the rate you maybe have a neo-functionalization. And then sort of using these sort of 4 parameters as ingredients, sort of plugging them into this QBD structure, and basically you get some sort of fairly giant matrices. So I think he was having fun trying to kill MATLAB with matrices of size 70,000 by 70,000 or so it's kind of fun. So other things that we've looked at, so kind of stability conditions of these models. So unlike the stuff Conrad was talking about with the gene tree situation, we're assuming you can't ever go extinct because your genes are doing some function that you need, and so they're essentially protected by selection at the level of of species. So you can't go extinct. You can go sort of supercritical if duplication dominates your 2 different kinds of loss of coding region or regulatory region, so things can blow up. But they can also be stable for some choices of your parameters. So Jehal's sort of characterised sort of those situations. And so similarly to what we were talking about with species trees, we're interested in computing basically the probability that conditional on having n tips, do you have, what balance do you have? So sort of I on the left. So basically by putting things in this QBD framework, we've got, I guess, access to some of these algorithms that people have, sort of general algorithms that people have dreamed up. Of course, we're discovering that, in practice, when you try and turn something from paperware into actual working, MATLAB code, and you have matrices of size 100,000 by 100,000, things can not work. But we're making very good progress, I think, on actually being able to compute these quantities here, and we think that we will be able to do that and characterize that. Essentially, use the sort of Laplace transformations to be able to get the sort of stationary distributions for these sort of quite complicated QBDs.
Yeah, and may I add to it also that the likelihood is something that we definitely can easily compute, because when we handle trees, reconstructed trees, there's no issue with the size of matrices, so we can easily compute those.
So basically, where are we at? We can kind of simulate under a wide range of kind of complicated, hopefully somewhat mechanistically motivated models. We can compute a lot of quantities of interest for the species trees, getting closer to be able to do that with gene trees. And then, so the next problem on our list, it was a bit of a title of aspiration, I guess, was putting these ingredients together and saying, can you actually come up with a gene tree, species tree reconciliation that is sort of aware of sort of sub-functionalization and kind of incorporates that into the sort of likelihood of different reconciliations. So,
and may I add? Barbara, I just wanted to add one more little thing. So I highlighted here generalized MBTs. Essentially, generalized MBTs is when a parent gives birth to more than 2 children potentially. So we're going to have that as well.
Oh,
thank you.
Thank you, Malgorzata and Barbara. Are there any questions?
Just a quick one. Are these matrices that you're trying to manipulate, are they particularly sparse or anything helpful like that?
The QBDs, like, they have a lot of blocks of zeros, but the actual blocks that aren't zeros are not particularly sparse in Jehal's case. So,
okay, Joyce. All right.
Any more questions?
None in the chat.
Mike has another question.
Another question, and it occurred to me when you were talking about the sort of a bias in speciation rates, and we've seen a whole lot of, this is a very left field question, a whole lot of talks about consensus methods as well, and I just wonder if anybody, perhaps yourself or anybody else, might have some thoughts about how we might get a speciation asymmetry aware consensus method from what you're doing, perhaps.
Yeah, Yeah, I haven't
Just
really
too bizarre a thought.
haven't thought about this so much in the context of consensus. Yeah.
It's sort of related to finding, the multiple gene tree, species tree reconciliation. So it might be worth having to think about. It might emerge anyway.
Right. Thank you.
Okay, so our next speaker is Chris Burridge, also from University of Tasmania.
Thank you. Just getting used to your mouse.
Oh, it's a terrible mouse. Don't call it mine.
All right, thanks for the introduction. Today I'll be presenting some work in collaboration with John Waters at the University of Otago. And this is the sort of talk where you have a tree now, what can you do with it? We were interested in predicting diversification rates in fishes and whether that related to migratory status. So first of all, we've already heard speciation and extinction quite a lot. What is diversification? For some people, they think diversification is speciation, but it is actually the difference between the two. And it's an important parameter to have some knowledge about because it determines the number of species that exist at any given time, so that's biodiversity. It can also tell you whether your biodiversity is going to increase or decrease through time. So a lot of people look at diversification and they say, can we predict what influences diversification? What traits are important? And traits relating to movement are often investigated in this regard, but they're kind of interesting in that movement can have a contradictory or can have contradictory impacts on diversification. So when people think diversification, they usually think of speciation. So the first thing is with respect to movement, isolation is valuable or useful. So species that are more sedentary or sessile should have higher rates of speciation and therefore higher rates of diversification. However, it's not all over if you happen to be a vagile species, if you move around a lot, because if you move around a lot, you might be more likely to find a new environment, you can undergo adaptive divergence and speciation. Mobile species tend to be more abundant and have wider ranges, so this reduces your chance of extinction, which therefore also increases your rate of diversification. And if you're more abundant, you typically have higher levels of genetic diversity. And genetic diversity is kind of the building blocks of evolution and speciation, and can also be handy to slow down your rate of extinction. However, we don't know the relative sizes of the pluses and minuses in this table. So our question was to try to get a handle on that. And we studied migration as a proxy for movement. Geneticists often equate migration with gene flow, but an ecologist will tell you, no, it's what whales do. Okay, so migration is the predictable and repeated movement between locations for the purpose of breeding or feeding. Okay. But the key thing here with respect to movement is migratory species are often more mobile than closely related non-migratory species. So we're using migration as a nice sort of proxy. You move a lot, you don't move a lot. So our question relates to migratory fishes. Do migratory species have higher or lower diversification rates? Okay, so the fish in question are galaxids. These are native to the southern hemisphere. They sort of look like salmonids. Salmonids are not native in the southern hemisphere, however. We've got 82 species, 14 of which are migratory. The group is widely represented in temperate environments in southern Australia, New Zealand, Patagonia and South Africa. And the adults of all species occupy rivers. And they don't really differ in a lot of features other than their migratory status. So we've got a nice sort of controlled system, I think. So if you happen to be a migratory species as an adult, you'll swim downstream and spawn in an estuary, and then your larvae will go out to sea and they'll spend between 3 and 12 months drifting around in the ocean. They'll then come back to the river and mature into adults. All right, so what did we do? We needed a tree, obviously. We'd previously studied the phylogeny of this group. However, we supplemented that with some data from new species. So it's not essential, but it's ideal that you have a fairly strong taxonomic representation from your group, and since our earlier study there was like 16 new species described, so we added them in. We built a tree using fairly standard methodology. We had some nice temporal calibration points. You'd like to know the sort of chronology in your tree to do this analysis. It's almost essential. And then we had our migratory status was coded simply as 0 if you don't migrate and 1 if you do. All right, so to investigate this question, we use something a bit like BUSY, but it's got a little addition, and that's called a hidden state. So the zeros are non-migratory states, 1 is migratory, and this is our null model. So we're not seeing any difference in speciation rates and extinction rates, whether you're migratory or not. However, we've got this hidden state, the A's and the B's, and that's providing us with a more realistic null model. So it's saying we observe migratory status, and that's what we're interested in, but there could be other traits or other features that we haven't observed that are also important for understanding diversification rates in this group. So we've got a hidden state in there as well, but in the null, no difference between speciation and extinction, whether you're migratory or not, but you may differ with respect to your hidden state. All right, so our test model, no hidden states, but you now can differ in speciation and extinction rates depending on whether you're migratory. If this model comes up better, you then typically entertain other models where you throw hidden states back in just to see whether there is something within either your migratory state or in your species with the migratory state that could also be explaining variation in diversification rates. Okay, so we've got a tree. The red species on this tree are migratory, but if you eyeball that, you probably don't have much of a clue as to whether they have higher or lower diversification rates. So let's jump straight to the best model. So our best model did indeed entertain different rates of speciation and extinction depending on whether you were migratory or not. So we have higher rates of extinction overall in our non-migratory, but much, much higher rates of speciation. So overall, our non-migratory galaxids have higher diversification. So this model has the hidden state within the non-migratory galaxids. That's telling us that there is something else in there contributing to variation in in diversification rates. The good news is both groups have elevated diversification rates relative to the migratory species, so migratory status is indeed important, but there is something else going on in there. We can have a look at our tree, and here we've got some coloured bars that are indicating tip diversification rate on the tree, and there's a scale break there to pay attention to. So our migratory species, very low rates of diversification. Our non-migratories have faster rates. If your tip state is predominantly assigned to the hidden state B, you are really, really fast with your diversification. Whereas if your tip state is predominantly assigned to hidden state A, you're still fast, but you're not quite as fast. All right, so that's them there. So in summary, non-migratory galaxids exhibit higher diversification rates. They have higher extinction, but much, much higher speciation. And among the non-migratory galaxids, it appears we have two groups with respect to diversification rates, but I'll not talk about them right now at least. So coming back to our little table, because our non-migratory species have higher rates of diversification, the only way they can kind of win that competition is if genetic isolation is really important for diversification. So the isolation of populations in species is really important for the diversification of these groups. In contrast, whether you encounter new environments, whether you're abundant, or whether you have high genetic diversity seem to be less important. All right, so we're not the first people by any means to address this question with respect to migratory status. Some other people have looked at it with a range of taxa. Some people have found the same results as us when they've looked at a much larger number of fishes, so that's Tedesco. Some other people have found completely the opposite, that our migratory lineages actually have higher diversification rate, including one interesting study here by Karush that's actually analysing exactly the same data set as Tedesco. And then finally an example where migratory status doesn't seem to matter at all. So one of our aims was, we wanted to understand the importance of movement and migratory status, and maybe we could use that knowledge to predict diversification. So that's not sort of a glowing, encouraging assessment or observation. So then we started to think about that a bit further, and it's like, well, hang on a sec. Yes, there are some species that are migratory and some that aren't. But when you actually look at the migratory phase amongst species, it's quite, they differ quite a lot. So in our galaxid example on the left, the migratory phase is this little larvae that washes around in the ocean for a few months. Whereas in other species of fish, the migratory phase is an adult with well-developed skills of locomotion and homing and everything else. So maybe it's not surprising we see differences amongst studies. The other thing we considered was the spatial context of the studies. So obviously you've got migratory species and non-migratory species, but what's the spatial environment in which your study system is operating? How far apart are habitats? What are the dispersal capabilities of species? So in this little graph here, we've got speciation rate on the y-axis, not diversification rate, that's fine, and dispersal ability on the x-axis. And the theory goes that to speciate a lot, you want to be occasionally founding a new population. So you're moving to a new place sort of occasionally, but not frequently. If you never move to a new place, well, that's really hard to speciate. If you're always moving to new places, well, that means as soon as you get there, you're probably still going to be exchanging genes with individuals from where you've just arrived from. But if you kind of occasionally found a new population and then you rarely genetically connected with your source population, that's good news for speciation. So where am I going with this? So for some study systems, your non-migratory species may hardly ever occupy or colonise a new environment, and your migratory species may occasionally cross that threshold, and hence migration could be good for diversification or for speciation and then diversification. Alternatively, in your study system, maybe it's your non-migratory species that happen to be able to occasionally cross some barriers and therefore promote speciation, whereas your migratory species, they cross barriers too easily and therefore they always maintain a genetic connection. All right, so when we look at our little study from the southern hemisphere with our galaxid fish, we've got a whole bunch of fish that live on these continents that are pretty isolated from one another. So we suggest that perhaps their migration, the dispersal ability of migratory species is sufficient to keep them all connected within a continent, but they might rarely be able to colonise new continents. Whereas this other sort of study with contradictory results to ours by Kurush, most of the species in that data set are kind of tropical or temperate northern hemisphere. And in these environments, the physical isolation of freshwater systems kind of varies more continuously. So it's perhaps more likely that species can find this sort of sweet spot of intermediate dispersal under which speciation can be high and therefore diversification as well. Okay, so in conclusion, if you don't migrate, you diversify faster if you're a galaxid fish. But we also think that perhaps the outcomes of studies of this type depend on attributes of the migratory phase rather than just you are migratory, and also the spatial context of your study system. Thank you.
Thanks very much, Chris. Any questions?
Yep. Nick.
Yeah, great talk, Chris. With the Kurush paper, was there differences in methodology, or were they quite similar to?
Yeah, Kurush used the HISI model, so differed predominantly in the size of the data set. Another interesting difference there is their data set's a lot bigger, but the proportion of migratory species is really, really small. So we were kind of like saying, we've got 17% and you've got like 0.5%. So that's another difference. There's contradictory results with the same number of species, and that was the Tedescu study, but rather than using kind of a HISI framework, they were looking at, let me see if I can remember this correctly, the percentage of migratory species within a clade and testing that relative to the number of species in the clade. But that also was somewhat arbitrary in terms of, so what are you going to call a clade? Is it a genus, a family, or something else? Mike Charleston.
Can I have a follow-on question from that?
Yeah.
So it seems like, I mean, the jury's still out on what the best methodology really is to understand these dynamics. So do you have a sense that that's a far off resolution, or do you think there's are we converging towards a right method of doing this? Or
in terms of things like HISI versus other things?
Yes.
Yeah. Yeah, Barbara actually asked me this recently. I think this was all kind of a new area to me, so I did a bunch of reading and which methodology, and certainly BUSY has some issues in terms of, you know, not being able to accommodate the hidden states, and there's other things perhaps in your data set that are important that you're not aware of. HISI to me is obviously an improvement in that regard. It's been tested relatively well in terms of simulations. So at my level, I'm reasonably confident with that. But yeah, there's certainly a whole bunch of debate in the literature, and it's, it's no different to, well, it's superimposed on, are you confident your tree is correct?
Yep.
Branch lengths and divergence time
I imagine
estimates.
data are going to be even more rare though, so.
Yeah.
Thank you.
Okay, let's thank Chris again.
Albert, are you there?
So our next speaker is Albert Sowong-Sono from the University of Tasmania.
Yeah, you ready to share your screen?
Can you see my screen now?
Yep.
Okay. Right. Yeah. So hello everyone. So thank you for having me in this session. So for these sessions, I'm going to talk about the shapes of phylogenetic trees under phase type distributed times to speciation and extinction. So actually, some of the stuff here are already in the poster session yesterday, and also in the paper that I'm currently working with Professor Barbara and Associate Professor Magujara. So for this one, I'm going to talk about three different stuff. So the first one is that the constructions of the model where we apply the phase type distributions to the speciations and extinctions process. And then the second one is that we are going to explore the different tree shapes under the model that follows the phase type distributions. So by tree shapes, I mean that we're going to explore about the balance, which is a degree, which is a measure to for a degree of symmetric in your phylogenetic tree, and also for the study on the distributions of branch lengths, which we can tell about the diversification rates in a given phylogenetic tree. And also for the last one is given that we have an empirical reconstructed phylogeny. So in this case, we're going to use two examples. So the first one is the squamate reconstructed tree, and also the angiosperm, which is a group of flowering plants, and that do the goodness of fit test for the model that follows phase type distributions and the other distributions such as Weibull and the exponential distributions. But for this one, we are going to assume that there is no extinction process assumes. So in the other talk earlier, we already have the equations for the extinction event as well, but for this one, we only consider for the speciations. All right. So for the macroevolutionary model that use phase type distributions, so phase type distributions basically it models the time until you reach the absorbing state in the continuous type Markov chains. So you have your state space defined where you have, say, m number of transient states, and your m+1 is your absorbing states. And so these phase type distributions is defined by this parameter alpha, which basically records the probability of starting in any of the transient states. And also you have this subrate matrix that records the transition rates of going between the non-absorbing state or the transient states. So you can think that biologically as the time until you see the speciation event or extinction events. the time you go between the phases in your phase type distributions as like the time spent by the species going through different phases until the species all goes extinct. Like in the previous talk that where we have like phase 0 and phase 1 from your individuals. And so we can think that the speciation process as being symmetric. So Barbara has discussed about the symmetric speciation. So following a speciation events, assuming that you have a binary tree, both children descending from the parents, the times or the lifetimes are drawn from an independent and identical phase type distributions. Right. So for particularly, we are going to use the structure of phase type distributions known as the Coxian phase type distributions. So in Coxian phase type distributions, you're only allowed to start from the first state. So that's why your alpha parameter has 1 in the first positions, and this is the structure of the subrate matrix. And this is the diagram assuming that you have 4 states in the phase type distribution. And so to study the effect of the model parameters to the diversification process, or in this case, the speciations and an extinction process, we consider these 2 examples that we found. So the first example on the left, that corresponds to the model where you have decreasing rates on either the speciation or extinctions. So what I mean by decreasing rates is that the rates of getting absorbed, if you are in the earliest states is larger than compared if you are in later states, and that's the opposite if you consider this example 1.2. So that one corresponds to a model where you have increasing rates. So we're gonna do simulations of 300 trees. Each tree has 100 alive tips, but we consider both symmetric and asymmetric speciation just for comparisons and for some choices of XY parameters, and we just assume set parameter to be equal to 2. So we're gonna do in 2 steps. So the first one is where we assume the times to speciations are drawn from the phase type distribution to check the effect of these model parameters on the speciation process. But the times to extinctions are drawn from exponential distributions with a constant rate. And for the second simulations, we reverse the process. So now the times to extinctions follow a phase type, while the times to speciations follow exponential distributions. So in order to study this balance on your phylogenetic tree and the branch length distributions, we use this statistic called the beta statistic, and also the gamma statistic for the branch length distribution. So in the earlier talk, we have already other measure to quantify the tree balance. So that one given by this di given n. But for this talk, we are going to just use the beta statistic. So beta statistic basically tells you the degree of symmetry on your phylogenetic tree. So if your beta is negative, you tend to have unbalanced tree like this figure on the right. But if your beta is positive, then you tend to have a balanced tree. While if beta equals to 0, that's consistent with the Pure-Bird process or the Yule process. And also for the gamma statistic, if you have gamma negative, that indicates decreasing speciations. So that means that in your phylogenetic tree, you tend to have shorter branches if you are closer to the roots compared to if you are closer to the tips. While if your gamma is positive, that indicates increasing in speciation rate. So you have branch length that tends to be shorter towards the tips instead of towards the right. So these are just some figures from the simulation. So the figure on the left, that's where you suppose that your times speciations are drawn from the phase type distributions. So for the balance as measured by this beta statistic, you can see there are some changes with just some 4 different pairs of X and Y, but we don't see that change for the gamma statistic for the branch length one. So that indicates that your tree balance is mostly controlled by the speciation process, but not the extinction. As for the second simulation, that's where your times to extinctions, we use the phase type distributions, while the times to speciations are drawn from the exponential distributions. So in this case, you see the opposite case. So for this one, the branch lengths are affected by the extinction process, but not the balance on the phylogenetic tree. Right, so that's the first one. So the second one is where we try to use the empirical trees. So in this case, we assume that there is no extinction process, and also we assume that we have independence on branches, and they are the times until you see each speciations are drawn from a same phase type distributions. So in phylogenetic tree, you have two different sets of branches. So you have internal branches, and also you have external branches. So in this example, in these three examples, you have four internal branches. First, we have independence. You have this product here, and then this comes from the density function from a phase type distributions. While the for the probability of seeing that external branches, you can think that as being the probability that the lineage is the
fan coil on top of, if I don't mistake that, on MS1 and MS2, they're all VRVs. Yeah, no, that's all right. You know, I'm in the same I'm in the same boat as you, getting to know the buildings around. But just for the filters, you know, I had a chat with we have a team meeting every month with.
I see some other.
We have a team meeting every month with you guys, you know, so Daniel Labert and Tom, and I've actually suggested to Daniel to, you know, if I was easier for you guys to do, you know.
Yeah. Okay. Yeah. Sorry. So the probability of seeing these external branches, you can think that as probability that the lineage is survived at least until time t, so that you can just use the cumulative function from a phase type distributions, and then you have this product of independence. So then the probability of seeing that particular tree with no extinction involved is given by this. So basically just find the parameters from phase type distributions, assuming that you have branch length as your data that maximize this quantity here. Right, so these are just some fitting from the squamates and the angiosperm tree, and then we consider some clades from those trees, and you can see that the one that use phase type distributions, they give the lowest AIC value, and you can see from the delta AIC here, compared that if you use the exponential distributions and variable distributions, except for the last one here where we consider this clade. This one, the variable one, performs better, a little bit better than the phase type, but that might be because of the number of branches used are not many. And so for the angiosperm tree for the flowering plant, we also see the same trend. So this one we consider this general Coxian phase type distribution as well, other than these two examples that I mentioned earlier. And that phase type distributions also has better fit plates and also the angiosperm tree. So that's the so just to summarize my talk. So we can see that different, just by using some parameter choices on the beta statistic, in phase type distributions, you can see that we have beta that is negative, beta that is positive. That corresponds to you have tree that is unbalanced and also balanced. And also we see the effect of the model parameters on speciations and extinctions process on your phylogenetic tree. And also we see that using branch lengths from the squamates and angiosperm tree, we found that we use phase type distributions that provides better fit than the other distributions, viable distributions. I think that's all. Thank you.
I apologize for that interruption. It was somebody who had directly dialed into the video conference room. It took me a moment to find them and hang them up. My apologies. You kept your composure extremely well.
Yeah, well done.
Thanks very much, Albert. Any questions? Anyone's in? So I have a question. With the beta statistic, do you know what kind of distribution of that you get with your Coxian pH model?
Oh, you mean like how the beta statistic looks like when I use the phase up distributions?
Yeah.
So essentially, like you can get beta that can range from negative to positive values. So that suggests like is like this model that follows phase up distributions. If you just choose some of the choices, you can have unbalanced or balanced tree. But that's good because if you want to fit with the empirical trees, that means that you possibly can find parameters from the phase up distribution from this model that use phase up distributions that fits with the shapes of your empirical. Hope that answers your questions.
Yep, thank you. Any other questions? Okay, let's thank Albert again. And our next speaker is Joao, who's also from University of Tasmania.
Yeah, sharing my screen. So, is it the sharing screen is fine?
Yeah.
So, yeah. So hello, I'm Jia Hao, and I'm currently studying in China, so I only can perform here. And so my talk is about the substantiation model of the gene family evolution, but its balanced gene state, which Barbara and Laura have mentioned before. And so for today's talk, I will just get into 4 sessions. And the first part I will introduce the gene structure we use. So we'll consider gene structure consists of coding regions and regulatory regions. So in this case, we have 1 coding regions and 3 regulatory regions, and each regulatory regions can perform the function. And so if a gene loses the coding region, it also means that this gene is dead. And also if the gene loses all the regulatory regions, the gene is also dead in this situation. So in the gene family evolution, we will have 3 main events, which is subspeciation, which means that occurs when at least 2 long mutation knock out different regulatory regions. And I will give details later for this subspeciation. And non-functionalization, it means that it just loses 1 gene. And also new functionalization is 1 copied may acquire a new function. So we will get extra function in the gene family. This means 3 main events is showing in the 2 genes cases. So thanks for Tristan have published this paper, and we have cases like that, which we start at 1 gene and 1 is the copy of the gene. So if a non-functionalization occurs, it means that we probably lose 1 coding regions, and then this gene is dead, and the copy of the gene will perform all the function. And so also this is the case which we lose the first function in the first gene, and then we lose the coding region. So it's still non-functionalization event. So this is all the non-functionalization. It also can occur when all the function is lost. So this gene is also dead. Only the another gene will perform all the function. So this is also non-functionalization. And so for the some functionalization, we will show that both genes will survive, but only each gene will perform, each function will only perform in 1 gene. So in this case, that is the subfuncionalization. So, but in this case, we haven't included the new functionalization. And if we consider new functionalization, we will create a new function which is not just performed like that. And also, in this process, we will have a selection protect the function, which means that at each function at least will perform in one string, so we will not lose any function in the process. So let's move to the family of strings. So we will not only consider two strings cases, we will have a family of string. In this case, we have a rate, which is we call it UD is the rate of duplication, and UF is the rate of new functionalization, and UC is the rate of losing the coding regions of a string, and UR is just losing a function in the regulatory regions of a string. So I will present the detailed binary matrix model, which can record all the information for the gene family. So in here, we have AIJ, which is record just 0 and 1. And if a 1 in the matrix, it means that there is a function in this string. The function is still survive. And if 0, it means that this string have a new non-mutation occurs in this function. So in here, we have a 3x3 matrix, which the row will record the number of strings. So we have 3 strings here and 3 columns. It means that we have 3 regulatory regions. So each will perform one function. And in here, because the second row, which we only have a 1 in the third column, so which means that this string will be protected. So we wanted each function at least performed in one function in one string. So we will have 3 strings, but only 2 redundant strings, which means that only 2 strings can be lost, which is the first string or the third string can be lost, and the second string will be protected, and because this function will be protected. So the total rate of this matrix A for leaving the current state, which is have lambda A, and for here, because we have 3 strings for the previous example, we have 3 strings, and we have 3 times UD. That will be the total rate to edit a copy of rows. That's the gene duplication. And also because some of the gene will be protected, so only the redundant gene can be lost. So we will have total rate of losing any of the n redundant genes. So we will lose the whole row. That will be the rate of that. And also here if we consider the new functionalization, then each gene can be obtain a new function. Then we will edit the extra columns and give another column with 1, which is this string acquire a new function. And also because the process can be lose some function, so that will be the total 1 in the whole matrix, and minus the MPV. It means that the column will only have 1 1 in there, so that one will definitely will be protected by the selection. So we'll just minus that part of 1 which is protected, and then the rest of function can be lost. So here is also an example how we construct a tree using this detailed binary matrix model. So I will explain the deprecation first. So at first we have 2 genes. So we have a chair, which is gene 1 and gene 2. And in the first process, the red road have been deprecated. And then we got 3 roads, 3 times 4 matrix. And so the first one is being deprecated. So we'll get a cherry with 1 and 3. And after that is the third row is deprecated. So we get a 4 by 4 matrix, and then we have a cherry with 3 and 4. So that is how the deprecation can generate a tree. And then for gene loss, I think we can start from the matrix which from the previous slide. So at the first gene loss, we lose the second row. So our gene 2 is lost. And then the second step is the in the first thing is third row, but in here will be from the second row, but it can be recorded as the 3. So then we lose the second row and we got loss for the gene 3. And in this case, we also can record it the bench length, because for lose the gene, we can lose the coding regions, and also can be lose the function, which we can lose 1 by 1. So for the total rate, we can record it the bench length for each gene tree. So from here, we will get to reconstructed tree, which we will not record any extinction. So we only have the tree which recorded the gene survival. So what I have mentioned before, that is the reconstructed tree. We can do the tree balance for that. So in the long run, we can think about the new functionalization rate is still, which we will not add any extra function. You have the upper bound for that. So for the tree balance we use the beta statistics, which Francois Albert have mentioned that the different value of beta can be percent the tree shape is unbalanced or balanced lot. So the simulation for here, we will have a stable process, which is UR plus UC is greater than UD, and then there will be have a upper bound for the number of genes, and the beta value we got, we have 2 stopping criterion, which is one is running time, and the second will be the first time to reach the number of leaps. And the beta value we got is more balanced for the tree generated by this under these parameters, which UI is 0.8 and UC 1 and UD is equal to 1 with this stable process. And for unstable process, we have 2 possible cases. One is we have UR plus UC equal to UD. And this special cases is if the number of gene and number of redundant gene is the same, then we have the same rate of deprecation and loss of the gene. But most of them is because we have some function and some genes is protected, then in this case we will have a higher deprecation rate. So and in this case, we gotten the beta is a slightly unbalanced. So for another unstable process, we will have UR plus UC is less than UD, then definitely the deprecation will be dominate the whole process, and which we gotten the beta value is very close to 0, which is very likely to be a uniform distribution ranking tree. So also it's just the U model, which is very similar to that. And also we have measured of the diversification rate. We're using the gamma statistics for the tree we generated, and to look at how the root can be close to the ancestor. So if the gamma can represent the tree state, then if we have a positive gamma value, it means that the internal node will be more closer to the ancestor, and for the tips, then will be have a longer bench length. And so we also have the simulation about how our model can shows the diversification rate. So as you see here, so if for positive, so in this area, it's more likely to be a stable process. So the UR plus UC is less than the UD, the 1. So that will be a higher value of the gamma. And then if the process is very stable and we have a higher loss rate, then the gamma will be just close to 0. So that will be all for this talk. Thank you.
Thank you, Zhahao.
Yes.
Are there any questions? Any from Zoom? So I saw you got different beta values for the different processes. Do you have any, have you looked at any real world examples and whether you get similar kind of values to what you did in the simulations?
Actually, that would be our further work because furthermore, we will try to use different value of the parameters and try to see how we will fit the real data set. So that is not working right now, but just that is what we try to do later.
Yep, okay, great.
Yep.
Let's thank Zhahao again. And the last speaker for Philomania 2020 is Tristan Stark from UTAS.
One of these two.
I was just going to share it from here.
Oh, yeah, I suppose that will work. That will work. Awesome.
Yep. Okay, so my name is Tristan Stark. Thanks for the introduction, Jonathan. So I'm a postdoc here at UTAS working with Barbara and Malgorzata, but what I'm talking about today is work that I did while I was doing a postdoc in Philadelphia at Temple University with David Libeles. And this work here is largely the work of Rebecca Kaufman, David and myself. And when I left Temple and Rebecca finished her master's, it's kind of sat around for a bit and we're sort of trying to pick it up now. So we've got a couple of new students on doing some of the work, Ryan and Maria. So what we're doing is we're looking at gene duplication, like you've heard about in the last few talks, but through a different sort of lens. And we're sort of looking at applying population genetic models. We know a lot about population genetic models. There's, classic results going back, to the 30s and 60s and things from Wright and Fisher and Kimura and Moran and others about fixation probabilities and inferring allele ages and all of that sort of stuff. But what we're wanting to do is test whether a particular gene that's sort of recently undergone a gene duplication event, whether that duplicate copy is associated with some selective effect. And so instead of looking at this at the sort of species level as we're doing in other work, this is looking at a single species population level. So what we're going to do is present a straightforward sort of hypothesis test, essentially, that's in a frequentist kind of framework, where we can kind of test whether a gene that's recently duplicated based on its proportion in the population is likely to be evolving neutrally or under selection, given that it's, it exists for us to have detected. So we've heard a little bit about gene duplicates. They're, generally understood to be important in establishing genetic diversity. This is partly due to the redundancy effect that Jiahao mentioned, which allows for mutation to sort of search the space of possible alleles with relaxed selection constraints. But, if it finds something good, so to speak, then selection is going to come in, and we would like to detect that. So there are a few mechanisms we have to consider with this. I'll just be brief. Jiahao has kind of detailed this. So subfunctionalization is where the functions are split up between two copies of the gene, or more than two potentially. So my little hand-drawn diagram here is my rendition of Jiao's diagram he had from our early paper. But you can see, both of these genes are going to be required to retain the full function of the original gene. And then there's neo-functionalization, where I've got this nice shiny new function that makes me, selected for. And the last sort of process that we think about here that hasn't been introduced so far in these talks is this dosage balance. So the idea here is that duplication can affect the expression level of some gene, and that can then affect some physiological function. Maybe it produces more metabolites or some such thing, and that can then have a selective effect. So the typical sort of picture of gene expression is this kind of asymptotic function where, depending on the level of gene expression right now, some physiological function, if you're in this sort of region of the space, if you get another copy and you get more gene expression, then this sort of abstract function is going to do more, and therefore it might be selected for. And alternatively, if you're in this sort of saturation part of the space, then the gene duplication is going to do nothing implicitly beneficial. And the last sort of thing is pseudogenization, where failing something to keep both copies around, one of them is eventually going to end up becoming a pseudogene through repeated null mutations. And so we apply this multi-allelic Moran model, which is one of the very classic population genetic models, similar in a lot of ways to the Wright-Fisher model, but I have a preference for it personally because of its relative sort of conceptual simplicity. You don't have to worry about all this counting. And so basically the idea is that we assume that a locus that had a single allele duplicated, and we consider this for both a haploid and a monoecious diploid population. And after it's duplicated, now there's two alleles, one being one copy, the other being two copies. And so we associate some fitness FD with the case that you have two copies, and assume that the fitness is 1 if you have one copy. And then individuals are just uniformly chosen in continuous time to be replaced, and the replacement allele is chosen proportional to the total fitness in the population associated with that allele. And then we allow for a duplicated copy to be neo-functionalized at some rate mu(n), having fitness Fn, or to become pseudogenized at some rate mu(p). And in our model, we sort of consider this equivalent to having just a single copy of the gene. You've got one that still works, so that's close enough. So this defines a continuous time Markov chain, and in fact, it's a QBD. And its transition rates are basically, I'll just show a couple of them here. The states here, I and J, I is corresponding to having a duplicated copy, but it hasn't neo-functionalized, and J is corresponding to your duplicate copy has neo-functionalized. So you can get an extra duplicate copy if you lose a wild type single copy individual and replace it with a duplicated individual, or, and I've kind of left this off here just to keep it real simple, but, or you could lose a neo-functionalized individual and replace them with a regular duplicated individual. And, you know, the same thing can happen in reverse, basically. So we have this death term and then this birth term, and here this death term and then this birth term. And there's also this extra little bit for the pseudogenizing mutations. And similarly, you can get an extra neo-functionalized copy through the neo-functionalizing mutation. But, and yeah, I've kind of left the details sort of under the hood there, but we also assume that neo-functionalization happens at most once. So you end up with this population model that, in this case here, you know, we always assume you start with one duplicated individual, and then as time goes on, a swap occurs. Time goes on, maybe a neo-functionalization occurs. And so in this example, we're transitioning through states 1, 0, 2, 0, and 2, 1, and then going on and on. And just a note on the interpretation of FD, which I think is not as obvious as the other things. So the case FD equals 1 is the sort of saturation case from that, that gene expression level graph that I had before, where having an extra gene that's doing nothing for you, you already had plenty of whatever it's coding for. The case where it's greater than 1 is the undersaturation case, where this particular product is limiting in its whatever pathway it's in, so getting more of it kind of cranks that pathway up. And alternatively, although this is a somewhat less sort of common case, one would think, it might have a negative effect, because, you know, it could just be that producing more of this metabolite, maybe you're a very small organism and spending your energy making something that is doing nothing for you is wasteful and therefore selected against, or maybe it ends up binding to something that it shouldn't when it's in these high concentrations, or, you know, it could be negative, basically. Or rather, the fitness effect could be less than 1. Selection could be negative. So also, generally, we think Fn is going to be bigger than Fd, because neo-functionalization, by its very nature, is the, you know, is selected for. So the main idea that we have here is that what we can do is we can condition on the survival of the duplicate, and then, you know, say you see one of these recent duplications in the wild, you can, based on this model, you can calculate the proportion of duplicate copies you would expect to see segregating at any time, and you can calculate a associated prediction band. So you can say 95% of the time, the proportion of duplicates, if they're you know, following this model, should be in this region, and that sort of defines a hypothesis test. And if you do this for the neutral case in particular, then you have a, you know, a test for neutrality, that if the proportions that you're observing are outside of this region, then you can, with whatever significance level you like, you know, reject the hypothesis that this is a neutral duplicate. And we can do the same sort of thing for the non-neutral cases, which allows us to see it's kind of a measure of power, essentially, that we can see how much selection we would have to sort of expect to be outside of this band. And here's some preliminary results that hopefully you can just about see. So we have here these blue lines are the neutral case, and these yellow lines, the expectation of proportion of population carrying the duplicated copy, that is, and these yellow lines are the expectation under some other selective regimes. So, you know, going down the page here, we're increasing the selective effect of having a duplicate in the first place, and going across the columns, we're increasing the selective effect of having a neo-functionalized copy. You'll notice, though, that here we have just n equals 100, and so this is sort of what remains to do here, where we're picking this project back up to make it, you know, a little bit more publishable. Directly computing probabilities under these models is you have to exponentiate the generator matrices of these huge, you know, huge matrices, like millions of columns and rows, which you know, is obviously problematic, and especially bad for the diploid case. I've kind of talked about the haploid case, but we're doing both. Turns out it's not so bad if you just consider the neutral case, because if you set the neo-functionalization rate and pseudo-generalization rate down to 0 and the fitnesses all to 1, then suddenly this is just a symmetric random walk, and that's, you know, easy. So we can get this hypothesis test, but only assuming no pseudo-generalization. So what we want to do is to use a diffusion approximation, as Conrad talked about earlier, to be able to take this n to large values and get the same sorts of results for large values of n, which is probably how I'll spend this weekend now that this is reignited and we really want to get it finished. So thanks to both Temple University for funding me while I was actually working on this, and UTAS for funding me now, and the Centre for Computational Genetics and Genomics at Temple and NSF for funding, and my collaborators, Rebecca over here, David over here, and Maria and Ryan, who joined the lab too late to be in a live group photo. So that's it from me.
Thank you, Tristan. Any questions?
We have a question from Christina. Christina, do you want to put your Zoom on and ask your question yourself?
Yeah, hi. Yeah, so that was more about the previous talk. So I was wondering whether you had also looked at other measures of tree balance, like combinatorial balance indices, and whether they're kind of, do the results agree, or how much does it matter which index or which measure of balance you use?
Yeah, I think like I've looked at the other measure of balance that you mentioned, like the Sakins and the Collis index, just to see like the model parameters from the phase distribution affecting these balance measures. So I think like when I try to look at that, they also and then compare with like some empirical trees, I think like they kind of get a good match as well. But I think like we chose the beta statistics because the other measures like Sakin and Collis, they need to be like normalized because they depend on the like the size of your tree as well. So that's why like we just stick with the beta statistics.
Yeah. Yeah. Also, in my case, for the gene trees, we have different size of tree if we consider of the running time. So we definitely Collis and Sakins need to normalize it. And but if we have a very large size of tree, then normalization is not good. That's I have tried also in the previous simulation, so I haven't put that the results in the talk.
Okay, thank you.
I'd like to add to it too. Can you hear me? Oh yes, I'm audio. So yeah. So another reason we like this beta statistic is that those probabilities that if you have a tree with n tips, I will be on the left subtree. We can actually calculate theoretically. We can derive theoretical expressions. So based on the models that we've been working on, we can so like for example quasi-birth and death processes or Markovian binary trees, we can write those expressions directly. So we have essentially like distributions of those, not just like running simulations, for example. So you can potentially fit parameters to your data for some model and then derive those expressions using analysis, using algorithms and analysis. So that's what's interesting and useful about using I guess beta statistics or comparing our expressions with the formula that uses beta statistics, which is something what I discussed in my talk with Barbara.
Okay, thank you.
Okay, let's thank Tristan first. And thank all the other speakers for the session.
And can we also do some extra thank yous?
Well, I wanted
Just
to,
some
first of
closing
all,
remarks? Okay.
I mean, I'm not going to give a big speech, but, you know, this has been an amazing 3 days. Thanks so much to everybody who came, who gave a talk, who presented, who stayed up late, who attended talks and asked questions. It's been amazing. We have 207 participants, which is by far the biggest Philomania ever. We had 48 presentations. And yeah, I mean, I've enjoyed it. It's been amazing, and I hope that everyone else has enjoyed it as well. So thank you to everybody. You've made it really, really great.
We sure Which brings me, of course, to a very important part of the conference, in which we thank our glorious organisers. I think back to when we decided it would be a fun idea to have, you know, have wonderful postdocs organising the conference. And of course, we didn't know that the world is going to be afflicted by a global pandemic and that the normal way the conference run would have to change entirely. And so I really, really commend you guys for putting together something that's been, I think, a wonderful hybrid conference experience. I've really enjoyed it. Good work on co-opting Josh in to do the website. Now we can't go back to our like stupidly coloured HTML website. We've raised the bar too high. But I have a few presents just to thank everybody. So I judged that Venta probably lost the most sleep overall of the organising committee, and so therefore the nicest bottle of wine goes to her.
I just open it now.
to Nick for fantastic work on the social media angle and just general enthusiasm pulling everything together. Josh for the fantastic website, showing my prejudices about what postdocs versus PhD students enjoy drinking, and also for Mike, who put his usual wonderful effort into getting our superb logo and the program. Thank you very much.
Thank you very much. Stuff to say too.
Well, it's not just, you know, I mean, I definitely want to give Josh a big thank you because the website's been amazing. He's also been just general tech support and, you know, sanity checks for me. Barbara, your expertise in putting the scheduling together was invaluable. So, you know, Nick and I had a look and we did some things, and then I did some more shuffling, and then I looked at times, and I really, you know, with your expertise, I think we made a pretty well-rounded program. Jeremy also has been a source of great advice and sanity checks for me. So, you know, the whole group has been all participating and helping.
Absolutely.
And thanks for that. That's lovely.
Yeah, so thanks so much. Yes, then we've got Mike. To give
I want to say a few things as well, if I may. So I'm sorry, I've got more things to say. And so I just get to jump in and talk now, which is kind of easy. It's good, cool. So I put a few We've already thanked people, so there's going to be a little bit of that, but anyway. It's been such a pleasure, again, to be at this conference right here in Tasmania, and it's been noted that Tasmania is a pretty great place to be right now. So next time maybe we'll have more of us here. We've been forced to go virtual, but it's meant that this year we've had this amazing large crowd. We've been able to welcome colleagues from everywhere, basically. As Svent has just said, over 200 participants, 90 countries, God knows how many time zones. I particularly enjoyed not having to run it this year. So thank you again, please, for running it. You two have been amazing. Let's give them a quick round of applause. And thank you also for leaving me just the fun bits, which is the messing around with the LaTeX and the graphics, so it's appreciated. If anybody's figured out the significance of the font, the Philomania typeface, do let me know. I don't think anybody's mentioned it yet, so just have a look. Scientific conferences like this one are the absolute lifeblood of academics for staying sane and happy and productive. This is the best part of my year. It's absolutely the most fun part of my job, getting together at conferences, scientific conferences, and actually conferring with my dear, wonderful colleagues and watching everybody else doing the same. It is just the best thing, and it keeps me going, and I think it probably keeps a lot of us going. It's worth noting not only how fortunate we are to have jobs just now, but also our great fortune to have such interesting jobs and wonderful, talented, and lovely people to work with everywhere. And even though we aren't sadly able to physically be together this time around, I want to say that Ventura and Nick and friends and colleagues and helpers have managed to pull off running a conference that has actually felt like the real in-place thing, with the new collaborations, the new ideas, the great conversations, even the beers in the pub. So that was amazing. Thank you, thank you, thank you. Something we do every year, of course, is hand out completely whimsical and utterly pointless prizes. This year, the prizes are even less valuable, being entirely virtual. However, the recipients of these prizes will be receiving handcrafted virtual Philomania mugs if all goes according to plan. So one of the traditional prizes that we dish out is, of course, the most tenuous connection to phylogenetics, which I think this year goes to Arndt von Hessler, who's also shown great ability to welcome new technology into his life through the use of green screen technology and a stick. So thank you. Well done, Arndt. For the best, kindest, most patient, exemplary parenting skills while still giving a coherent talk, I think Stephen Crotty should be also sent a virtual pat on the back. We'll turn that television off right now. For the most professional and polished recorded talk that many of us likened to the heightened levels of professionalism of a weather presenter, to Olga Chernomor, who put the rest of us to shame. We couldn't pick a best student talk. We're sorry. They were all an amazing standard. So to all of the students who presented, fantastic job, really well done, super. The most inconvenient time for a speaker award was a three-way split between Michael Hendrickson, Cassius Manuel Perez, and Mohammed Adesanya, sorry for mispronunciating that. I think it was all about midnight for their talks for them, wasn't it? So well done, you guys, for staying awake.
I I do have to say there was a little bit of discrimination there. If there was somebody that I knew had small children, I didn't schedule them a late night slot. So there was some discrimination going on in a positive way.
In a positive way. And for the best use of basement art and continuing contribution to phylogenetics, to Joe Felsenstein. What happens next year? Well, next year we're actually hoping to be, able to travel again. And I will be back to having some more involvement with it. But now that I've, seen what a wonderful job that other people can do, maybe I won't need to just do the fun LaTeX and graphic stuff. It will be number 13, speaking of graphics. So if anybody has some phylogenetic suggestions as a connection to the number 13, please let me know ahead of time. Phylomania 2021 will also be co-run or partnering with Australasian Bioinformatics and Computational Biology Society annual conference, which is going to be run here in Hobart next year too. And that's a bigger conference. And so much in the same way as we ran Australasian Evolutionary Society conference with Phylomania a couple of years back, we'll be doing that with Abacus. So that's exciting, and that was announced yesterday, and yours truly is the person who stuck up his hand to organise that, so yay. However, we will keep this hub idea running, I think. It's been very effective to run virtual sessions, so I think we'll have some kind of a hybrid conference for that as well, hybrid sort of sessions for that as well. However, if you can come and are willing to offset your carbon footprint by doing so, or sail, and we would love to see you here. And I think that is all I have to say. We'll also be coordinating with a molecular ecology meeting, Chris Burridge, which runs every year. And so we'll have the 3 meetings that won't clash. The current Abacus just clashed with us because, they're bigger and we're small. But anyway, so that's all. Thank you very much. Thank you very much for letting me have a few words.
Oh, thanks very much for that, Mike. Have you got anything else, Fenta? Or?
No, only we already have a suggestion. Thanks, Michael. Friday the 13th for next year's Phylomania.
Friday the 13th. Okay, thank you.
Done already.
Nice.
In real time, really. The only other thing I've got is that it's lunch, of course. At 1 o'clock, we made a booking, so I hope to see as many people as possible there. We've got a booking for 15, which should be enough. But it's at the Hanging Gardens. Look it up if you don't know where it is. I've never been there, but I've been told it's super cool and good food and good beer, so which is what we need.
Okay, well, we will farewell our participants who are not in the room. Lovely to see you all, and hopefully next year we will see you in person.
Bye. Thanks so much for joining us. Bye.